What I want to say first is that this is a great honor and privilege for me to be here. I'm actually representing a broader team, over 200 employees at Sarepta throughout the United States. I have a handful that are here today, scattered throughout. We'll be here through the week, so please make sure you introduce yourself if you don't know us, if you have any questions about Sarepta. We're here to help in any way we can. It's also my honor to present on behalf of our Chief Medical Officer and Interim CEO, Dr. Edward Kay, who many of you likely know. He sends his regrets as he's unable to make the meeting today, but I hope I can do this presentation justice. So the first slide is an eye exam for those in the back of the room. If you can read the last line, then you win. So we're a publicly traded company. I think everyone has heard of Sarepta. There has been a few news segments you might have read about. Essentially what I'll be doing today, this is just a legal slide, just to let you know that I will be saying some forward-looking statements about our clinical development program, the status of the eteplirsen new drug application, and many other aspects of our timelines regarding our clinical trials. They are subject to risks and uncertainties. What I would say is that if you have any questions about this, you can always refer to our SEC filings, in particular the ones from the end of 2015, our 10K, as well as our most recent 10Q that was filed at the end of March of this year. So that's the legal explanation. So before I get into the agenda that I wanted to present today, I want to make sure everyone is on the same page with respect to the eteplirsen NDA, the new drug application, and our status. There's nothing new to report today, but again, just to make sure everyone knows where we're at. On June 6th, we have communicated broadly to the community that the FDA has requested additional data from an ongoing clinical study entitled Pharm.D. This is what we're considering one of two confirmatory studies for eteplirsen if it is granted accelerated approval by FDA. What they requested is that we review the Western blot, do Western blot analysis on 13 biopsies obtained from boys who have participated in this trial. We'll be comparing baseline values to week 48 values. What we hope to do, we aim to get this information to FDA over the coming weeks so that they can make a prompt decision on the status of the NDA. So what should you do? One thing I want to acknowledge is that I've been with the company for a little bit under a year, but I've worked in industry for almost 20 years, and there's nothing that I've seen like I saw at the advisory committee and the support from the advocates. Without your help, without the patients and the families, and most importantly, the boys who are participating and the men that are participating in our trials, we would not be here today speaking about Sarepta's clinical development program. So we really, truly, truly appreciate the tremendous support. So please keep in touch. What we'll be doing, obviously, as a public company, when we have any information, we will be providing that information broadly, an update regarding the NDA. So we will do that as soon as we have information, and we'll be communicating directly to advocacy organizations such as PPMD, Jet Foundation, and the like. So please just keep your ears open. You'll likely know it as soon as we can communicate. But we always try to be very transparent. One of the things that I want to acknowledge is that although I'm heading up medical affairs and patient advocacy for the organization, there have been many people who have done so many things before I joined the company. So I definitely want to acknowledge that. I might have this fancy title, but without the groundwork that was done by the company over the last few years, we definitely wouldn't be here. And they've always tried to be very transparent, which I've really appreciated, and I enjoyed working with this team. And specifically, we want to make sure that if there are any questions, reach out to us. We may not be able to answer all of those, specifically because of the sensitivities around our NDA and clinical development programs. However, email us, call us. We will do our best. Another thing, again, I'm not even on the agenda yet, so I think I have about 2 hours. What we want to confirm to the community is that we are dedicated to developing potential treatments for Duchenne. You've heard about the NDA for eteplirsen. We've also announced recently that we are expanding our clinical development program for exon skipping for boys amenable to exon skipping 45 as well as 53. I think this is a trial that's been in the works for many months, if not years. So apologies for the delay, but I think we're at a point where we understand how that trial needs to be implemented. And I'm going to go through that in very great detail and make sure that I answer any questions about that trial, because it is going to be a very difficult trial and one that we're going to need the support of the community more than ever. The other thing we're going to do is continuously evaluate follow-on exon programs, and I think one of the things when I joined the company that I realized immediately was that the company that, Ed Kay, the CEO and CMO, he wants to bring treatments to all boys amenable to exon skipping and beyond that. So we want to, based on our ability, primarily based on finances as well as supply and grow, and then the need from the scientific need to determine safety and efficacy in multiple products. That's how we'll implement our clinical trials. So I'll go through the examples of the eteplirsen and the 45 and 53 programs. We're also going to be continuously looking for opportunities to expand into our next generation exon skipping treatments. Some of you may have heard of our PPMO platform that we've been working on. That's something that we want to expedite as soon as we're able to do so. Finally, but not of least importance, we want to collaborate to find ways that these exon skipping products, as they become reality, can also be used in combination with other therapeutics to really optimize clinical outcomes for patients. We're also not just dedicated to looking at exon skipping platform. We're continuously evaluating to determine whether or not other platforms such as gene therapy would be appropriate. Now to the agenda. I think Lauren Vogt yesterday, if you were at her talk, she did such a nice job to really explain the importance of genetic testing. So I'm just really going to briefly go through that. But what I would say is that PPMD has been a true partner and really has been leading many aspects of advocacy in important programs such as Duchenne. I'm also going to make sure that the audience understands exon skipping in a very simplified manner. I hope I can do that. I think there's a lot of folks in here that have much experience in our clinical trials as investigators, study staff, but I also realize PPMD, Ryan in particular, has been able to get many new families here today, so I want to make sure that you understand really the concept around exon skipping. Then I'll get into the clinical development program and finish off with conclusions. So really, one of the things that I thought was really interesting from the outset when I joined the company last summer was that with rare disorders, including Duchenne, the challenge always ends up being diagnostics. And with Duchenne, all boys end up getting diagnosed. However, there's an opportunity for these boys to get diagnosed earlier, as well as to ensure that the proper testing is being performed. When I review the Bushby guidelines from 2010, as well as the FDA guidance that PPMD helped lead or led the development of, it's very explicit that genetic testing is something that should be done. And again, Lauren had done a great job yesterday explaining the various reasons for that, but it really is now the current standard of diagnosis for many reasons. We have a great team back at Cambridge and across the United States, and we hope to expand into Europe, including the UK. What we have learned, what we try to do is we want to understand how can we help. And the only way we can figure that out is where are the gaps. So this just illustrates what the marketing team at Sarepta has been able to clarify as a real need, and again, relates to the importance of genetic testing. So you can see here that the data that we've collected, this is data on file at Sarepta, really about half of the population in the United States has not been tested, genetically tested. On the left there, you can see the bars really explain that genetic testing is typically done more often in the clinics where more patients are being seen, but there still remains a gap. So you can see on the tallest bar there that only 69% of patients in clinics that they see over 100 Duchenne patients have been genotyped. So again, just underscores the importance of genetic testing and really the gap that currently exists, and it could be for many reasons that Lauren explained yesterday. It could be the availability of genetic testing at the time your boy was diagnosed, or other reasons, access due to insurance issues and the like. One other thing to point out is that we've had multiple advisory board meetings over the past year in which we've engaged with Duchenne experts as well as Duchenne treaters. They've been treating Duchenne patients for many years. When we inquired about the understanding around exon skipping, we've had many physicians believe that mutations on exon 51 or deletions of exon 51 are required for eteplirsen to be amenable or applicable to those patients. So again, even if genetic testing is done, the ability to interpret the results is just the second step that's really important to do. So again, I won't belabor the point because Lauren did such a nice job to explain all of the reasons that genetic testing should be done, but one of the most important programs that I've been working on with PPMD, but they've really been, obviously taking the lead on it. If I can get this slide to work. Is the Duchenne, Duchenne program. And one of the things that I think Annie Kennedy had clarified to me early on, I think she asked me the question at a meeting in Europe. So Dana, what do you think the most important thing about this program is? And my first inclination was that it's free of charge genetic testing. But she made it very clear, and I truly understand that it's much more than that. It's the ability for PPMD to help out with the interpretation and as well as genetic counseling, which I think is very important. So even if genetic testing has been done previously, I think what Lauren said yesterday was make sure you have that piece of paper and that you understand what it means. And if you don't, or if your physician does not, contact PPMD, they can help out. This is just an example of one of the templates that they would hand out to healthcare providers that explains if out-of-frame deletions are seen on genetic testing results for patients, amenability to exon skipping can be determined quite easily from this example. So let me get into exon skipping and really go through that to make it really simple. There we go. So Dr. Nelson had really done a great job to explain the importance of understanding genetics. So what I'm going to do is apply that to the theoretical potential of exon skipping and the mechanism of action. So first, dystrophin protein again acts as a molecular shock absorber and protects muscles during contraction. As you know, boys and men with, we'll just leave it on there, but boys and men with mutations on the DMD gene, many of them have out-of-frame deletions. And these are really the target for exon skipping. What we're trying to do is to restore the reading frame to allow for the production of a shortened, truncated, but functional dystrophin protein. The next slide that you saw briefly, here it is again the exon map. So the largest gene in the human body, 79 exons. This, essentially the way that really to think about this is that it's like a jigsaw puzzle. The pieces need to fit together in order for dystrophin to be produced. So the whole concept of exon skipping is around these out-of-frame deletions. So you can see here, this is an example of a deletion of exon 49 and 50. And if you look at 48 to 51, they do not match up. And this is where the reading frame doesn't allow for read-through so that dystrophin can be produced. Our PMO technology targets a specific exon and allows that to skip. And you can see here, it's skipping from 48 to 52, allowing for the reading frame to be allowed for reading of the reading frame by the ribosome so that dystrophin is produced. Again, shortened functional dystrophin. So that's, again, a very simplified approach to exon skipping. It is more complicated than that, but it just gives you a general concept around what we're trying to do. So what I would ask is, what is wrong with exon 51? This is an example of a patient that would be amenable to exon 51 skipping. So there's deletions in exons 47 through 50. The answer is nothing. And that's the point. And again, one of the things that we had learned from the market research that not all clinicians had understood that. And we're trying to do our best to further the education. And again, PPMD is a great partner to access to good not only for free genetic testing, but for that interpretation, so you know what it actually means. Whether it's exon skipping, nonsense mutation therapies, and this is not being self-serving. There are several companies that are now very interested, as well as moving into the clinic with exon skipping therapies. So it's not just for Sarepta's programs, it's for all programs, as well as programs that we don't even know exist yet that will be coming down the road. So that's why the importance of genetic testing really ties into this specifically, but really it's a broader reason to get tested. So what I'm going to do now is just make sure that everyone's on the same page with Sarepta's Duchenne clinical trial program. I think there's a lot of focus on study 201, 202. Everyone's heard a lot about that. That's what we filed our new drug application with the FDA on, the database on that. That trial is now up to 5 years. And again, I would say that on behalf of everyone at Sarepta, we're extremely grateful for the families that participated in that trial, the boys that have participated in that trial over the years. It really has been an incredible partnership to make sure that we can get the data that we need to understand whether or not these products work. So this just, I know this is hard to read because there's quite a bit of information on here, but there are 6 active clinical trials and 1 planned. I'm going to get into the details of some of these trials, but what I did notice in the packets that were provided by PPMD, there was a brochure that goes through several, if not all, of the active or ongoing or planned clinical trials. So we'll show you the status of those trials and provide a lot of information around that. So please refer to that as well. I think the story around Sarepta and our program is complex. There's no doubt about it. However, I think what we've learned through the years, again, the 5-year clinical trial, just reaching beyond 5 years this year, study 201 and 202, we've learned quite a bit, but we've also worked in partnership with the advocates in particular. Pat, during her speech yesterday, and pretty much every speech that I've heard from Pat, she always highlights the importance to ensure that clinical trials, and then at the end of the day, if treatments are approved, that they're available to all patients, to all boys and men affected. That's what we've tried to do, and I think there's a balance that we always need to consider. We want to ensure that we're in the broadest population. There has to be a reason that we do that. There has to be a scientific reason. In this case, gathering safety and efficacy data in a broad population is absolutely very important and supportive information to determine, yes, is this product safe in a broader population? Is it efficacious? So we've tried to do that. This just illustrates the broad range of patients, and I would say to all of those companies that are starting to develop programs, you might not want to use the exact study designs. However, what you want to do is really consider how can you get to the broadest population in these clinical trials to get meaningful data, whether it be efficacy or safety data. And that's what we try to do. We're doing that as well with exon 45 skipping and exon 53 skipping development. So you can see here there's quite a broad range of patients that boys and men, that we plan to enroll or have already been enrolled in some of our clinical trials. As I mentioned in the previous slide, there are about 135 boys or men with Duchenne that will ultimately be enrolled in those trials. For the 45 and 53 skipping products, it will be about 140 based on the current plan. What I want to do is get into the details around this ESSENCE clinical trial. I mentioned the PPMD trial. The PPMD trial is the one where FDA had asked for additional data, the Western blot data, and that's what we're considering one of two confirmatory studies for eteplirsen if granted accelerated approval. ESSENCE is the second. I'm going to get into the trial design. There's been a lot of communication within the community and from Sarepta to the community about this trial. I know this trial has actually been in the works for quite some time, but we plan to have this actively enrolling this year. So, as an overview, we intend to enroll 100 patients, and although we realize this is going to be a difficult trial, we need the community to help to enroll this trial as soon as possible, work with the investigators and study staff, contact Sarepta. We'll answer any questions that you have. The way we set this trial up is it's a 96-week placebo-controlled study. It was changed from 48 weeks, and I'm going to explain why in a couple slides. We've also built into the trial a rollover to open-label extension for 96 weeks. The randomization will be 2-to-1, so meaning that for essentially 66 patients, approximately, will be randomized to receive treatment, either active treatment, either SRP 4045 if amenable to exon skipping 45, or SRP 4053 if amenable to exon 53 skipping, or placebo. So again, about 100 patients in total. We also plan to conduct this in Europe, in the US, Europe, as well as Canada. For the inclusion criteria, another change that we made was the age required for enrolling into this trial. It was originally planned for between the age of 7 and 16 years of age, and again, I'll go through the details as to why we changed that. And then again, amenable to, again, it goes back to the genetic testing and the need, especially to determine whether or not you may be eligible or your boy may be eligible for this trial. So you need to know whether or not you're amenable to skipping 45 or 53. And what we'll be doing is entry will be based on the average 6-minute walk test, 2 consecutive days between 300 and 450 meters. And their patients need to be on a stable corticosteroid dose for a minimum of 24 weeks prior to entry. Things to consider. What we realize this is a difficult trial, especially a 2-year placebo control. So what we've done is, again, try to ensure that the randomization allowed for the least amount of patients on placebo as possible. We've also built in an interim analysis. What we'll be doing is when 75% of the boys to enroll in the trial have enrolled, a data monitoring committee, external data monitoring committee will review the data and determine whether or not the trial can end early. So the earlier that the faster that this is enrolled, the more meaningful that is in terms of being able to potentially stop the trial earlier than 2 years. So this would be done at the end of the 48 weeks when 75 patients have been enrolled. Again, to point out, all boys will be eligible to enroll in the 96-week open-label extension, which could again begin earlier depending on the interim analysis. So what I want to do is really highlight the importance of this trial. This trial is critical to Sarepta's clinical development program and for future exon skipping development. So this highlights the 3 real key points that will come out of this trial and what we're trying to do. Again, number 1, this is considered by us one of 2 confirmatory studies. If we get accelerated approval granted by FDA on eteplirsen, because the chemistry backbone, the PMO backbone chemistry of these products are similar to eteplirsen and will help to support safety and efficacy of eteplirsen. Secondly, this is what we're considering our pivotal phase 3 study for both SRP-4045 and SRP-4053. So what that means is that our intent is to use these data to file for potential approval with the regulators. Lastly, but I think as important, is that we hope that these data will serve as a foundation for our follow-on exon programs. I think the company, for many years, has spoken about a platform approach. What we hope is that the 6-minute walk test data, the dystrophin production, and this trial, the data that we collect from this will be meaningful and help us to argue against, or for, I should say, our follow-on exon programs. So it's this is a very meaningful trial, and I'm going to go through some of the Q&A that we realize has been coming from the community. Probably shouldn't have those first few slides. Okay, so why double-blind placebo-controlled trial? So again, I think everyone who was remotely even involved in AdCom or attended AdCom understands the importance of the double-blind placebo-controlled trial. So we are following guidance. We also, again, believe it will serve as a foundation for our follow-on exons. And why did we extend it from 48 to 96 weeks? It's really based on the learnings from our establishing clinical study in which we did not see meaningful separation or statistically significant separation until up to 2 years. So we want to make sure that the worst thing we could do is conduct a trial over 48 weeks that doesn't have data that's meaningful. So we want to ensure that when we do this trial, it's our best chance at having a successful trial and meeting our primary endpoint. There's a couple more slides here. So why was the age range reduced from 7-16 to 7-13? We want to ensure that we enroll a very homogeneous population. Again, this was brought up at the FDA advisory committee for Tupperson regarding patients that they have a less predictable disease course, and that's what we've seen with patients over the age of 13. What we want to do, please, if there are any parents that have boys that are turning 14 that may be amenable and could potentially enroll in this trial, please, we want to make sure that we prioritize those patients that get screened as soon as possible. One thing to note is that boys have to be 13 at the time of randomization, and the screening period is approximately 7 to 8 weeks. So that's something to consider. So please reach out to us, contact your doctor, genetic counselor, be in touch. We're going to communicate directly with the community to make sure that this information is known and that as soon as sites are available to enroll patients in screen, that we notify the community at that time. So conclusions, we're committed to continuing our focus on development of our potential exon skipping therapeutics. Genetic testing is extremely important, as you've heard multiple times now. We have ongoing clinical trials. Please refer to the PPMD brochure, which will really help, but also contact us at any time. As far as the phase 3 Essence clinical trial, I've already kind of belabored the point of that. It's extremely important for the long-term programs for Sarepta. So again, the confirmatory study for Tupperson is the pivotal trial for 45 and 53, as well as helping us with the foundation for all exon programs. Lastly, but not least, is that, again, there's many folks that are working very hard at Sarepta that want to bring these therapies to these boys as soon as possible, and that's what we're working on. So thank you very much, especially to the boys and men who participated in our clinical trials. Thank you. Is there one question that I can answer? I'll answer yours too.
Hi, David. Thank you for the presentation. We appreciate it. For the last year, we've been in touch with Kara and Jenny from your team as well. And my son has the 45 skipping and will complete the exon skipping. What was surprise or obviously disappointing from our end to change the placebo to 96. So that is obviously difficult for us to consider as a parent after waiting for quite some time. A couple of questions for you. So the exon 53, the 51, sorry, what you're saying is that you did not see any significant change in 48 weeks, and that's your rationale. You essentially go to 96.
Yes.
At the same time, you expect parents to consider the fact that you will review data at 48 weeks for 45 if 75% of the boys have enrolled. Now, it's kind of contradictory to what you're saying you've already seen in 51. So obviously from our end, because the composition is the same, you may not see any difference. And if my boy was to enroll today, then he may have to wait 1.5 years until 75% of them enroll or have gone through 48. So it doesn't give us much confidence as a parent. Obviously, we're disappointed because of the decision.
Yeah, no, and we appreciate that. And again, this is not something that we want to do. We believe it's our best chance to ensure that at the end of the day, that we get the data that we need that would actually support potential regulatory approval, that would get to the broad population. But what I would say is that we don't want to play games and say this is my end in 48 weeks, because that's not true. What I would say is that yes, it's the same PMO chemistry backbone, but SRP 4045 and 4053 could be more efficient, and you may be able to see, especially we have a very tight criteria in terms of baseline 6-minute walk test and age. So we believe that by having a very homogeneous population, the reason why we have that data monitoring committee to look at 48 weeks is so that we could potentially end it earlier. But I would say that might not happen for sure. So that's why we're ensuring that it's built to be a 2-year study so that we have the best chance of a successful trial.
All I can say is that having an older child, this has put us as a parent in a very interesting bind, given that you have utrophin, which may come with an open label. There's myostatin that's going on, and as you know, it's really time sensitive. So I know it is our decision as a parent, but all I can say is that we are very disappointed.
Yeah. And we definitely understand that. And again, what we did was, again, I think everyone's pretty aware of our situation with respect to eteplirsen. It's now been 5 years for that clinical trial. We can't do that again. The community can't do that again. So I think we're trying to balance with what we believe at the end of the day will give us the most meaningful results, so that we could get to a broader population quicker and not have another 5-year trial. And it would be a shame to do a 48-week trial and the results show nothing. So, but trust me, I think if anyone knows Ed Kay, who's our Chief Medical Officer and interim CEO, this is not something that he wants to do. This is something that we believe we need to do based on many factors, including FDA guidance.
Thank you.
That was essentially my question.
Yeah.
Has there been any discussion with the FDA as to what criteria will be used to evaluate this at 48 weeks? Is it going to be something other than the 6-minute walk test, which you've already said, quite likely is not going to show a significant difference? Dystrophy distribution, will that?
Yeah.
Will there be something else other than the 6-minute walk test to evaluate it?
So the way we would be doing this, and I can't speak to our communications with FDA, but I can speak to how we're going to evaluate it, and it is based on 6-minute walk test, and it's sequential. So essentially, if 6-minute walk test does not look as though it has a significant difference at 48 weeks, then it would have to continue for 96 weeks. So again, we're not assuming that this will end in 48 weeks or longer, really, based on how fast this enrolls. But it is something that we built in in the event the tighter criteria as well as the efficiency of these products in terms of exon skipping, if they were better, to be able to do that. So again, we don't want to play games and say we're trying to do this because it could be shorter. We're doing this so that in the event that these look like they work better than 51, that we can end the trial.
And which level of dystrophin expression you expect?
Which level of dystrophin expression? That's something that I wouldn't be able to speak to. I think, again, we're in a sensitive period with FDA, and I think even at the AdCom, I think everyone had really heard about what's the meaningfulness of 0.9% dystrophin, and I think FDA commented that they're not sure. No one knows. So I'm not sure what the bar is, but what we're trying to see is that does this produce dystrophin like we've seen with the eteplirsen? So I think we have to move on, but we're here through Wednesday, and we have a booth up there, as well as a poster, so please stop by. We're happy to answer any questions that we can. Thank you.